Bismillahirrahmanirrahim. Assalamu alaikum. This is Exploring the Concepts, and I welcome you all in my today's video. Once again, I am with ninth class biology, and my today's topic is mitosis. This is from unit number 5, and the course is from FBISE and the BLD publishers. The textbook is of BLD publishers. So let's start the term cell division with the term cell divisions. Cell division is actually, you can see in this diagram that this is a single parent cell. On whenever it is going to decide that it has to make its copy, then it's need to divide, it's need to make a copy of its chromosomes present inside the nucleus. So first of all, it has to make a copy, then you can see a furrow deepen on its cell membrane, and then finally this further deepens and break it into two daughter cells. This is what is called as cell division, right? When one cell makes its copy, right? So there are two different types of cell divisions. Let's discuss the cell division. Cell division is actually the process by which a parent cell divides into two or more daughter cells. You can see in the last diagram that how one cell divides into two cells. So types of cell divisions as far as types of divisions are concerned. So there are two main cell divisions which are going on in the living cells. One of them is mitosis and the other is meiosis, right? So mitosis is actually, so the last diagram, if you can recall that diagram I've shown you in the first, the slide, that one cell is going to divide into two cells. So whenever the parent cell, if the parent cell has some specific chromosome number, like you all know that this is a species specific number, chromosome numbers are species specific number. So if a parent cell number has, for example, we are taking the example of human being, 46 chromosomes. If a parent cell has 46 chromosomes, then after completing the division, if the two daughter cells too have the same number of chromosome number, that is 46, it means this type of division is called as mitosis. Means the parent cells and the daughter cells are exact copies of each other. Means the two daughter cells are identical to each other, and they are the identical to the parent cell as well, right? So they are genetically identical. So look at the definition. You need to learn it. You need to write in the same way. You use black pointer to write the definitions. A type of cell division in which a cell divides into two daughter cells, each with the same number of chromosome as were present in the parent cell. अगर तो उनका chromosome number parent cell जैसा है, then it is called as mitosis. We have another definition or another way to say the same thing. Mitosis is a process of cell duplication in which one cell divides into two genetically identical daughter cells. So this is what mitosis is. It's a type of cell division, but the thing is, the daughter cells and the parent cell are genetically identical. Like all of the genes, they have the same number of chromosomes, they have the same type of genes. Every information is similar between the daughter and the parent cell. This is called as mitosis. Like for example, you often notice that you have got a cut on your skin, and then after a few days it will heal it, and you have got the new skin over there, which was same like that of the skin present before. So that what is the result of mitosis, right? So wound healing is actually because of the process of mitosis going on in our skin cells or wherever we have got a wound. So mitosis discovery. Who discovered this process of cell division, which is called as mitosis? He is a German biologist, Wilder Fleming. In 1880s, he studied some sperm cells, some egg cells, and he found out that there is going on a process, because we all know that we cannot see any chromosome inside the nucleus until or unless they are going through a dividing phase. So he studied a sperm cell in which there is a division will going on, and he find out the chromosomes, the condensed form of chromatin, and this is Welder Fleming who identified the process. Then properties. There are some specific conditions needed for the process called mitosis. First of all, it's a type of cell division. You all know that it's a type of cell division, and in which only nucleus has to pass through specific changes, right? It's a series of changes through which nucleus has to pass, and it is always and always occur in the eukaryotic cell. Prokaryotic cells, prokaryotic cell cannot go through the process called mitosis. It is going through a process called binary fission, right? So for prokaryotes, there is a binary fission process through which they can divide their nuclei, which is a simple process. While as far as eukaryotic cells are concerned, all of the cells that have nucleus and membranous organelle in them are called as eukaryotic cells. Like for example, our cells, they are eukaryotic cells. In multicellular organisms, the somatic cells undergo mitosis. You all know that's all of the somatic cells. One thing more important, although our germline cells are um, they are two the eukaryotic cells, but they can go for both the somatic and the meiosis. Another type of cell division can occur in those type of cells, right? So all of the somatic cells in our body, they only go through mitosis. Only and only mitosis can go through them. But germline cells, with this few exceptions, few of the cells have gone through mitosis, but rest of the cells will go for meiosis in case of germline cells, right? So properties as far as mitosis are concerned. So what is the need? First of all, the cell should be eukaryotic, the cell should be somatic. And secondly, mitosis can take place in both the prokaryotic and mitosis can take place in both the haploid and diploid cells, right? Whether the cell is diploid or haploid, it can go through mitosis, and but cell should be eukaryotic. Now, the phases of mitosis. Mitosis is a complex and highly regulated process. So we all know that it's a complex process. Obviously, as it is going on in the eukaryotic cell. Secondly, it has a very important responsibility. It has to form two similar cells, so it should be complex, and it has to be complex and obviously regulated as well. So whenever there is an involvement of nature, the things are highly regulated and highly systematic. So there are two major phases. The one is the karyokinesis. Karyo, the word karyo means, it's a Greek word. It is derived from a Greek word karyon, which means nucleus. So karyokinesis is actually the division of nucleus. Cytokinesis, the word cyto means cell, and cytokinesis is actually the cell division of cytoplasm, or the cell is called as cytokinesis. Now, the karyokinesis, right? We are concerning in mitosis, we are most of the time we are concerned with the karyokinesis. So, karyokinesis is divided into four phases: prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. You need to learn all these things. Better to learn through these slides because they are highly simplified. So, let's start with the very first phase of mitosis, that is prophase. Look at this diagram. It's a chromatin, right? Whenever there is a nucleus which is not going through a division, so the whole of the chromosomes, they are found in the form of chromatin network, a very fine thread-like structure, right? But on duplication, they get a copy of it attached with the help of a centromere. Whenever we see a centromere in the center, it means the chromatin is has go through a phase called S phase, right? And what about this? This is a condensed form of chromatin. We will see the genetic material that is in a loose thread-like form, chromatin, starts condenses into highly ordered structure called chromosome. Now, this is the highly ordered structure, right? That what is called as chromosome, and how it is formed by the condensation, by removing the spaces between the chromosomes. So, the basically chromatin, it coiled and keep on coiling on itself and forming a highly ordered structure, which is called as the chromosome. Such chromosome is made up of two sister chromatids bound together at the centromere. So, how would you identify a chromosome? It must have a centromere in the center, and it has attached a copy of the same same chromatin, right? So, it must have two chromatids, that means, because it has gone through a phase called S phase. Now, look at this diagram. This is the one chromatid, this is another chromatid. Both the chromatids are called as sister chromatids. They have attached in this place a centromere, right? Centromere is basically a region which has no specific gene, right? That is the region which is just need to join the two chromosome with each other. अब इन्हें क्यों join करना है? Because we do not want them to get separate, right? We want the copy of the chromosome to remain together. That's why they get attached through a centromeric region. And on this region, we have attached a binding protein called kinetochore, right? Centromere has a binding protein called kinetochore. Kinetochore, it's a very specific type of protein which is going to help the spindle fibers to get attached through the kinetochore. So every chromosome has, which you can see in the dividing cell, must have two chromatids attached to the centromere, and centromere has a specific protein called kinetochore protein, which is a binding protein. So this is how chromatin is found present inside the nucleus. They are highly, the in a thread-like structure, and these are the nuclear pore you can see. So they can take out a one thread of a chromatin. They are basically in this form. They get coiled on each other, and this condition we found in chromosomes. Right. This is animal cell micrograph. How condensation result in the visibility of the chromosomes. We are unable to see the chromatin, right? But in the form of chromosome, we can see it. And whenever we see that chromosome, it means the cell is going through a dividing phase. So each chromosome has kinetochore at centromere. I have already tell you about the kinetochore. Kinetochore is a complex protein structure where the spindle fiber is going to attach. Now, what happened next to the prophase? So we have identified up till now that the condensation of the chromosome should be the very first step. What is condensation? Condensation is actually the coiling up of the chromatin on its own on self. Now, duplication of centrioles. S phase will also, as far as S phase is concerned, S phase not only duplicated the number of chromosomes but also the centrioles as well. I mean the centrosomes as well. So there are two centrosomes in the dividing phase, in the dividing cell. So movement of centrosome to the opposite pole. So the formation of the spindle fiber has started in the prophase, as you can see in the diagram. So this is the the cell and nuclear membrane, the chromosomes in the form of duplicated copies, and these are the spindle fibers, and these are the centrosomes which are trying to move towards the opposite poles. And in the late prophase, what we have seen, the nuclear envelope is in the form of fragments. That means that the nuclear envelope get dissolved and nucleoli get disappear. This is the nucleoli present in the nucleolus, which is present inside the nucleus. It is also, we are unable to see here because it is to get disappeared, and these are the completion of the central spindle fibers as well. So movement of the centrosome to the opposite pole of cells, formation of microtubules by joining tubulin proteins present in the cytoplasm, and then formation of microtubules by joining tubulin proteins present in the cytoplasm. Formation of spindle fibers, microtubule formation of spindle, mitotic spindle. So अब होता क्या है यहाँ पे, हुआ ये है यहाँ पर कि सबसे पहले क्रोमोसोम्स कंडेंस हो गए. पहला स्टेप तुमने समझ लिया, क्रोमोसोम्स गेट कंडेंस्ड. Second step is the disappearance of the nucleoli, the disappearance of the nuclear membrane, right? It start disappearing, right? Basically dissolving. Then the division of the centrosome, though, that already has taken place in the S phase. And now the development of the spindle fibers, right? Development of the spindle, which is actually the arrangement of the microtubules in the center of the new cell. ठीक है, वो सेंटर की तरफ आके जमा होना शुरू हो गए, एंड तो बोथ द सेंट्रोसोम दे विल मूव. एंड ये कौन बताएगा स्पिंडल फाइबर्स को कि किस तरह अरेंज होना है तुमने, तो वो बताएंगे दो सेंट्रियोल्स. एक सेंट्रियोल एक कोने पे चला जाएगा, द अदर सेंट्रियोल विल मूव टुवर्ड्स द ऑपोजिट पोल, and then spindle fiber will arrange between the two centrioles. It is very important to note. This is the spindle fiber who is going to tell the microtubules to arrange in the specific order. So formation of spindle fibers by microtubules, formation of mitotic spindle, they are also called as mitotic spindles, right? Disappearance of, I already have told you about these things. So सॉरी, ये अब इस डायग्राम को देखो. दिस इज द chromosomes. You can see the chromatids, the centromere, the spindle, the centrioles, the spindles, and these are the microtubules. In the late prophase, metaprophase, they are trying to go to the next phase, completion of the spindle fiber formation, you can see. And in this diagram, starting from the G2 of interphase, There are two centrosomes. We can see in this diagram, pair of centrosomes are there, and the plasma membrane, sorry, the nuclear envelope is still intact. And there, this is how they are going to get copied and condensed. In this phase, we are unable to see that, and there is a starting of the initiation of the formation of spindle fiber, and then this is how the prophase ended up. Right, it is trying to arrange the the chromosomes on the center of the spindle fiber, which is also called as equatorial plate, right, or metaphasal plate. So this is what is called as the equatorial plate or the metaphasal plate. It is most of the time the center of the cell as well, but it is actually the center of the spindle fibers, right? So they arrange themselves on the center of the spindle fiber like this and like this. So every chromosome has its own copy attached with it through the centromere, right? So attachment of the spindle fibers kinetochore with the kinetochore. And now the most important thing, as we have already have studied this thing, that centromeres have kinetochore attached on them, and so the spindle fibers which are going to attach to this binding protein is called as kinetochore binding protein, kinetochore spindle fibers. So the kinetochore spindle fibers will attach to the kinetochores. ठीक हो गया? अब यहाँ से next is there are other polar spindle fibers starting from one centriole, ended up the other centriole, right? So they are called as polar spindle fibers. But those spindle fiber that has fall or the stopped at centrum kinetochore of the centromere are called as kinetochore microtubules, kinetochore spindle fibers, kinetochore spindle fibers, right? Attachment of two kinetochore fibers from opposite poles with each chromosome, arrangement of chromosomes on the equator. So that what is called as the metaphase. That we have ended with the metaphase. Now what happened at the anaphase? Actually, every kinetochore spindle fiber is attached with the kinetochore of the kinetochore of the centromere, right? They shorten. They contracted towards the centriole, right? वो contraction आ जाती है उनके अंदर, वो पीछे की तरफ खींचने लग जाती हैं, जिसकी वजह से सेंट्रियोल दरमियान में से कट जाता है, और हर डुप्लीकेटेड में से एक chromatid will move toward one pole, right? हर duplicated chromosome में से एक copy will move towards the one pole. Centromere divided and chromatids become chromosomes. Now they are called as chromosomes because they are no more attached with us at the centromere. So and they are now becoming the chromosomes. Spindle microtubules pull the genetically identical chromosomes, and there are two other genetically identical copies of the chromosomes. Let's for example, this cell has two pairs of chromosomes, okay? One pair and the other pair. So these two pairs, they are identical to both the cells. Half of the chromosome will move toward one pole, the other half will move toward the other pole, and all of them are actually the copies of each other, and all of them are actually exact copies of each other, right? That's why we call that in mitosis there is identical from the daughter cells are identical to their parent cells. What happened in anaphase? Pulling of kinetochores spindle fibers towards the originating chromosomes. So they pull towards the centrioles. Division of chromosomes, sister chromatids then pulled apart towards the respective chromosomes, and separation of the identical copies of the chromosomes. Separation of the identical copies. So one, this is chromosome number one, this is chromosome number one, and that was chromosome number one in the while they get copied. So then telophase. In telophase, we can see that there is a depression in the wall of the, in the wall of the cell membrane, and it get deepens, right? This furrow get deepens in the start of the telophase. There is no deepening. There is no furrow. We cannot identify any furrow in the start of the cell. But later on, we have developed for the telophase, we call it as the reverse of the prophase, the development of the nuclear membrane, development of the nucleoli, and the centriole, the removal, or you can say the dissolve ho jati hai spindle fibers, and they have developed their own cell by all means, right? Finally, this is called as telophase, when both the chromosomes, they will reach to the opposite pole, and they have developed every required thing, but still the furrow has to deepen further for the development of the two cells. Reversal of the prophase, formation of new nuclear envelope around each set of separate chromosome. Chromosomes unfold back into chromate. One thing very important is that once again the chromosomes will convert in the form of chromate. This is another. This is also called as the telophase. This is a mother cell, one of the mother cells which is going to go for mitosis. This is, it has passed through the S phase. Everyone has made its copy, and they attached through the, the centromere, and they are called as chromatid in this form, and they are called as chromosomes as well because we can see them in through the microscope now. This is the prophase, right? And this is metaphase when they attached on the metaphasal plate, and this is how they are going to move towards the opposite poles in anaphase. Every chromosome has the blue one blue will move toward one pole, the other blue will move toward the other pole. Means equal divisions of the chromosomes will take place in anaphase, and this is once again the two daughter cells form which are just like the parent cells. That is what is called as mitosis. They are forming the exact copies of their parents, which are which are similar by all means, the genetically and the physically as well. Then the mitosis in plants. Mitosis in plants is a little different as in plant cells. It has a large central vacuole, and the nucleus has to go towards one side. And plant cells does not have centrioles as well, as there is no enough space for the spindle fiber to get get disoriented. So formation of spindle fibers by tubulin protein, obviously spindle fibers will form, but centrioles are not needed for guiding the spindle fibers. So this is how plant cell and animal cell will form their spindle fiber formation. Prophase 1. Centrioles inside the centrosome. Centrioles are there, but there are no centrioles present in the plant cell. Please, plant cells does not have centrioles. They lack centrioles. The spindle fiber assembles without centrosome. There are some astral tubules that help the spindle fibers to arrange in that specific order. Spindle fibers are formed by centrosome, which we have already discussed. This is how they get, the division, cell division, cytokinesis. How cytokinesis will take place in case of plants. Furrow develop and deepens in case of animal cells, and there is no phragmoplast or the cell plate is formed in the animal cell. But in case of plant cell, the cytokinesis is totally different. As first of all, there is formation of the cell plate or the phragmoplast in case of plant cell. What is phragmoplast? Phragmoplast and how it is formed? These are actually the Golgi bodies, right? The vesicles from the Golgi containing the material for the formation of cell wall. All of the Golgi bodies, these basically structures are the Golgi bodies. They contains this material needed for the cytokinesis and needed for the formation of the cell wall, sorry. So all these Golgi vesicles contains the calcium and magnesium pactates, which are important for the formation of cell wall. So they arrange in the mid of the cell, and finally they join with each other. And in the next diagram, I will show you this. This is cytokinesis in the plant cell cleavage furrow and the development of the two daughter cells. This is how, first of all, they have arranged in the mid of the cell. Like in this diagram, you can see these are the Golgi vesicles. Finally, all of the vesicles, they join with each other and forming a continuous plate in the center of the plant cell. Like this, so this is the final structure of the plant cell, how they divide. So the cytokinesis in plants is totally different because they have to develop a phragmoplast. How phragmoplast is formed? By joining many Golgi vesicles. Golgi vesicle containing the material needed for the development of the cell wall. So Golgi body is basically defining the midpoint of the two cells. While in case of animal cell, it's very easy to form the contractile in the ring and then division of the cell into two. Cytokinesis in plants, you can once again you can see in this is the cell membrane on both the sides, and this is the Golgi vesicles. And these are the Golgi vesicles in the center. Movement of vesicles derived from the Golgi apparatus to the middle of the cell. Formation of the membrane bounded disc called cell plate or phragmoplast. Fusion of the plasma membrane with the membrane of the cell plate and its content join the parental cell wall. Formation of the two daughter cells bounded by its own plasma membrane and cell wall. So that is what is the cytokinesis in plant, which is very difficult and very specific to the plants, right? Okay, so that is all from my side about the mitosis. If you have any question, you can ask me. You can write me in the comments. So You can ask me any question. I can draw the diagrams as well for you if you want. And we will meet again in the next lecture. Allah Hafiz.